Hello everyone, this is Supreet Kadakol, Assistant Professor, Department of Zoology, Sri Venkatramana Swami College, Bantwad. Today, in this session, we'll see the syllabus for the final year, 5th semester, that is paper 5, Cell Biology and Biotechnology. So mainly, this paper consists of 4 units. So in that first unit is the Cell Biology. So under that Cell Biology unit, first 1.1, that is introduction, will be having history of cell biology, which includes the discovery of the cell, cell theory, and subdivisions of cell biology. Mainly how cell biology is influencing different many streams like biochemistry, microbiology, biotechnology. Along with that, we have scope of cell biology. So what all studies that happens in cell biology and what type of illustrations which were given by different scientists on cells and the type of working. So tools and techniques of cell biology, which includes different types of microscopes, ultracentrifugation. So here in microscopes, you will be studying how the microscope works, what are the parts of microscope, and what are the different types of microscope like ultra microscope, scanning microscope, electron microscope. And in ultracentrifugation, what is the purpose of use of ultracentrifugation in cell biology? Mainly it may be helped in extraction of DNA, RNA, proteins, etc. So in the second subunit, that is chromosomes, which includes morphology of chromosomes, structure. So depending on what structure or what type of chromosomes mainly they are present, like centromere, telomere, and difference between heterochromatin and euchromatin, and types of chromosomes, chromosome number, and what is genome. Under this, we will be studying ultrastructure of chromosomes and also different types of chromosomes like giant chromosome, polytene chromosomes, Lambrecht chromosomes, and nucleosome model, and supernumerary chromosomes. So in the third subunit, nucleic acids, very important subunit. Here we will be studying about different types of nucleic acids, DNA and RNA, what are their types, and introduction to the genetic material, or how the identification took place by different scientists or the different experiment that were given by Griffith, Avery, MacLeod and McCarthy, Hershey and Chase experiments. And while I was telling about the nucleic acids, you will be studying about chemistry of nucleic acid, what are the composition, and double helix model which are given by Watson and Crick, and mechanism. Mainly in your lower classes, you might have come across central dogma of molecular biology, which tells you that molecule which undergoes replication, transcription, translation. So all these three important mechanisms in detail you will be studying in this unit. So this is about the first unit of the cell biology. Whereas in second unit, the continuation of the cell biology only. In the first subunit, we will be studying about plasma membrane and cell junction. So we know plasma membrane, it is a semi-permeable membrane which helps in movement of solutes and solvent across the cell. And there are cell receptors. What type of receptors and what type of ligands which helps in movement of ions, mainly potassium, calcium, iron, etc. So with the help of receptor, what are the functions, what are the types of cell junctions. So in between two cells there is a junction that we call it as cell junctions. So it may be attached by different types of filaments, or it may be attached by different types of intracellular spaces. So that will be studying in this subunit. Whereas in second subunit, cytoskeleton and cell mortality, so here there are different types of cytoskeleton elements like microtubules, microfilaments, intermediate filaments, organization and function. So when these are combined along with these cytoskeleton elements, how plasma membrane is held through the tissues and cells, and along with the organs also. So those all the cytoskeleton elements will be studying in second unit. Whereas in third unit, the general characteristics of cell differentiation. So why all the cells are not equal in size, why different organs have got different cells, and what is the reason why different cells have different functions for different organ, and nucleocytoplasmic interaction, how nucleus will interact with the cytoplasm of the cell, and what is the purpose of interaction. So that all will be studying in cell differentiation. Whereas in the last subunit, that is cancer and carcinogenic agents. So this is also very important subunit in cell biology by unit 2, where we will be studying about what is cancer, what are the characteristics, what are the types of cancers, what do you mean by oncogenes, then what are the cancer causing agents, or we call them as carcinogenic agents. It may be by physical method or it may be by chemical, biological, and what are the therapies which helps in curing the cancers. And there are some techniques we use immune system to enhance the cancers or to degrade the cancer, that is immunotherapy, radiotherapies, chemotherapies. And what is the role of telomere in cell aging and cancer, and little about apoptosis. So programmed cell death, that will be studying why the cell division or why a single cell cannot be living for whole life, why it undergoes cell death. So that will be studying in this subunit. So these all are the topics which are covered in unit 2. Whereas in unit 3, cell biology, third unit, in the first subunit we are studying about the genetic code and protein biosynthesis. So here you will be having some hypothesis and different biosynthesizing cycles, where you will be knowing what are the different properties of genetic code, Ubel hypothesis, components of protein biosynthesis, mechanism, very important mechanism of protein biosynthesis. So how protein is degraded or how protein helps in formation of amino acids. So that will be studying in the first subunit. Whereas in second subunit, the cell division, which includes mitosis, in this we will be studying the complete cell cycle, phases of cell cycle, the interphase, prophase, then mitotic phase. Under that mitotic phase, you will be studying metaphase, anaphase, telophase, and also cytokinesis, and difference between mitosis in animal cell and plant cell. So if you have studied about plant cell and animal cell in detail in your lower class, so we can make the difference between animal cell and plant cell along with on their mitosis. So with this significance of the mitosis, why mitosis is required for cell division, and there are some inhibitors which prevent them in cell division, prevent them from cell division. So that is about the mitosis. Whereas in the third subunit, we will be studying about meiosis. So sexual cell division. So here will be formation of synaptic complex, mechanism of crossing over, and along with that we will be studying about significance. And there are stages in the meiosis, how when compared to mitosis, like metaphase, anaphase, telophase, prophase. Here along with that we will be studying about the significance, difference between mitosis and meiosis. So this is the third unit, which comprises of mainly cell division and the protein biosynthesis. So this is also very important unit where maximum questions can be asked. Coming to last unit, that is biotechnology, not much in detail but very important for your life science studies, which includes the first subunit, that is genetic engineering. Here you will be studying about the introduction of biotechnology and what are the different types of enzymes like restriction endonuclease 1, 2, 3, and the use of linkers, how they are mainly helped in recombinant DNA technology. For example, if you are having two different genes and they are having their own merits and demerits, if you want to make a merit of one organism, then you will go for recombinant DNA technology. So only the advantages of that gene you will be incorporating and insert into a plasmid or the bacteria. So that is how this type of work or this type of studies will be doing in the first unit, the first subunit of the unit 4, that is introduction. And along with that you will be studying about the genomic library, gene library, and the host cell, transformation and transduction, very very important, and the part electroporation, liposome mediated cultivation. So little about the cultivating different types of bacterias that will be covering in the first subunit. Whereas in second subunit you will be studying about the techniques. What are the different types of techniques we use in biotechnology or in overall life sciences, like that is PCR technique, which helps in amplification of DNA, southern, northern and western blotting. These are the blotting technique which helps in enhancement of proteins. DNA fingerprinting, very important for the criminological studies. What is the principle, what are the methods we use in DNA fingerprinting and the applications, and DNA sequencing. So we use different types of methods like electroporation, and also different types of genes we use, and protein engineering, fish, RAPD, RFLP. These are the important smaller subpart which we need to study about in the second subunit. Whereas in third subunit, that is application of biotechnology, so if these all techniques where we can apply, so mainly in pollution control, mainly in bioremediation, biotechnological disease prevention, and pharmaceutical vaccines, transgenic animals, then stem cells, gene therapy, monoclonal antibodies. So these are some of the applications which are mainly helping to overcome different types of diseases or pollution control, etc. So this is about your paper 5, that is cell biology and biotechnology. Thank you.